Before we start the podcast, we wanted to let you know about WestOS. If you're a teacher or pupil at a school in Scotland, then you now have access to hundreds of quality assured video lessons created by teachers to support your remote learning. You should be able to find WestOS within the app library of Glow, where you can add the tile to your own personal launchpad. Click on the tile and you'll have immediate access to stream lessons in every area of the Scottish curriculum, with video lessons already available for biology at National 5, Higher and Advanced Higher. Now let's get on with the podcast. This podcast is sponsored by Lecky textbooks, revision guides and practice papers, everything you need to learn, review and prepare for your senior phase assessments. Browse the books at www.leckyscotland.co.uk and get 40% off using the discount code LeckyPodcast40. You can now also collect Young Scot reward points by using the codes within the description of each episode to claim discounts and rewards on their website www.young.scot.
If you take a wee on the frog and then it starts producing eggs, then you're pregnant basically.
In biology, the terms conformer and regulator refer to two different strategies that organisms use to maintain their internal environment, or homeostasis, in response to changes in their external environment. In this episode of the Higher Biology podcast, we will speak to fishery scientist and former president of the North Atlantic Salmon Conservation Organisation, Professor Ken Whelan, and herpetologist Ollie Thomas, as we look to enhance our understanding of metabolism in conformers and regulators. Understanding the difference between conformers and regulators can help us appreciate the incredible diversity of life on Earth and the ways in which organisms have evolved to survive and thrive in different environments. We start by meeting Professor Ken Whelan, who's a science graduate from University College Dublin and a global expert on Atlantic salmon.
Thank you very much indeed, Paul. Delighted to be here.
Maybe you could tell us a little bit about yourself, Professor Whelan, and why you might be here.
Yeah, okay. I started really in terms of my interest in flowing water and things in water at a very young age, because my dad was a very keen sea angler. My brother and I spent a good bit of time then, long months indeed, with my aunt down in one of the counties along the east of Ireland, down in County Wexford. And my cousins, would you believe, were avid poachers. And because of what we saw them catch in local rivers, we started to shift our focus away from sea fishing to fishing rivers. And I really got fascinated with rivers. The whole basis for my career is really rivers rather than the individual fish or the individual insects or whatever that are in there. And we were very lucky because living just outside of Dublin City, we had a lovely little trout river, which is only about maybe 4 kilometres from where I'm speaking to you today. And that little trout river had a really progressive angling club. And in the early 1960s, they were actually running classes for juveniles, teaching boys and girls how to tie flies and teaching us how to fish for trout. So that was an incredible privilege. At the time, we didn't realize how lucky we were. But I just got, again, fascinated with the creatures that were in the river, particularly the invertebrates. And as a hobby, I used to go and search for these invertebrates, try and identify them. And we used to spend quite a lot of time cutting weed and so on in the river, really with a nefarious purpose in mind, because that made it easier to catch the trout. But the whole thing really just built to a situation where I became passionate about rivers and the life that's in rivers. And from that really grew my interest then eventually in fish and particularly in salmon.
When you weren't fishing in those early days, a large chunk of your time would have been spent at school. What was your favourite subject at school?
I think I'd probably got 3 subjects really that I was really interested in. I always loved English. That's always a great advantage because it gives you really an opening in all sorts of different areas. I also became really passionately interested in chemistry and also geography. A bit of a strange mix, but I have found in my career that those 3 subjects have served me very well. And there are 3 areas that I'm still very interested in.
So I guess young Ken wouldn't have expected to be on a higher biology podcast later in life if that wasn't one of your favourite subjects.
Absolutely not. And I'm probably not allowed to say this because it'll really get all your students whooping with joy. But I really wasn't very fond of school. School wasn't really a very inviting place in the 1960s. Certainly not for me. There was a lot of boring academic work involved. I was interested in outdoors. I was interested in biology. We didn't have biology on the boys' curriculum in Ireland at that stage. So my savior really was the chemistry, because my interest in chemistry gave me an opening then to actually start taking an interest in physics and maths and so on. That gave me the opportunity then to get a county council grant, which was the only way I could get to the university. And I managed to do that. But once I got my hands on the university, there was absolutely no stopping me. I was just in my element. So it was a strange sort of beginning. I mean, certainly you're right. If anyone had said to me at 14 or 15 years of age that I was going to end up in life as what people now call me, and I always find it very strange, an academic. I don't see myself, I don't feel that I'm an academic. But obviously, I have a real interest now in getting to the bottom of why things are as they are and what we can do to help things that are damaged.
Maybe you can tell us a little bit more. How did young Ken go from that classroom where he wasn't enjoying life to becoming Professor Whelan, the academic, as you're now known, as you say?
Again, it was a strange and very practical journey, really. I did quite well in my final degree. There was a very famous trout biologist in British Columbia, and I literally, out of the blue, wrote to him and said to him, would it be possible to do a PhD with him? And he said, yeah, he seemed very enthusiastic because of my interest in trout and so on. I wrote back and said, well, I have no money. And he said, well, that's a bit of a problem. So what he said was that if I did a master's degree very quickly, he was fairly sure I could get a teaching fellowship and that I could do my PhD in British Columbia. And I was about halfway through my master's when an ad came up for a biologist in what was called the Inland Fisheries Trust in Ireland. They were the group at the time that looked after recreational fisheries. And I kept saying, oh my God, can you imagine getting a job with these guys? I was 23 years of age. I didn't imagine in my wildest dreams that anyone was going to offer me a job. So I went for the interview, and myself and a very good fishing pal, we were offered the two jobs that came up because we seemed to suit exactly what they wanted. So I was then in a situation where I had to make a terrible choice. What do I do? So I decided that really, if I come back after 5 or 6 years in Canada, this is the job that I would have dreamed of. So I took the job, but I then had to set about then doing my PhD on my own over an 8-year period without any great support from the university, I hasten to add. And eventually I managed to put together all of the material and got my PhD. And interestingly, my PhD was not on fish at all. It was on mayflies. So I did a PhD looking at the potential disappearance of the big mayfly. In Latin, it's called Ephemera danica, which is economically very important to Ireland because it brings in a lot of tourists to the Midlands and to the Western lakes in Maytime. And because of eutrophication and enrichment of lakes, it was under pressure. So I was asked, could I develop techniques whereby we could restock the lakes with these particular insects if the need arose? And that was what I spent my time doing. I took over a part of a small hatchery, and I then looked at some of the old techniques that we used on the River Test in England, and I updated them. And I managed to develop techniques then whereby we could actually start to restock some of the lakes with these mayflies. And that was what my PhD was about.
And once you'd finished that, you ultimately end up as the research director for the Atlantic Salmon Trust.
Yeah, well, the PhD I did, as I say, while I worked, a little bit of an anticlimax, I must say, because when I went in on the Monday morning, having done my final interview for the PhD, it was, nothing had changed. I was still in the same job. I was still being paid the same amount of money, but it was just the satisfaction really of finishing something that I had started. So I was very happy in my work. I had a fantastic project at that stage where I was looking at one of the really big river systems in Ireland that had been damaged by big works, big peat works. And what we did in Ireland was our bogs were so extensive, or our peatlands were so extensive in the Midlands, that on an industrial scale, they used to harvest the peat to generate electricity. Shocking when you think about it. But at the time, of course, people had no idea of the impact this was having. But the environmental impact was very obvious on some of the rivers. So I was running a big project looking at the environmental impact of peat particles, some of the bigger Midland lakes. And I got a phone call in 1982 from my boss's boss. And we'd had a change in that the organization that used to be the Inland Fisheries Trust had now become a body that was more centrally placed within government. And we were given responsibility for eels and for salmon. And he said to me, Ken, would you like to take over the salmon portfolio? And I said, but what about my boss and my boss's boss? And he said, well, I talked to both of them and they reckon there's really nothing interesting left to do on salmon. And they suggested maybe you'd like to do it. And I said, yeah, I'd love to do that. So the very first job I got was to go with a friend of mine who made all sorts of promotional films for the organization, to go around the world and to basically look at salmon management in various areas across the globe over a 12 or 14-month period, and to put together 2 films to try and explain to people how salmon were being managed in other parts of the world and how we might do things differently in Ireland. So that was a fantastic start. And I still have contacts now that I made during that first journey to all those interesting places across the globe.
Our planet is full of interesting places where you inevitably find interesting people. On one expedition to South America, as well as meeting the indigenous Sani people of the region, I met a herpetologist called Ollie Thomas. And this seems like an ideal time to bring him in to give us a quick insight into herpetology and how conformers control their metabolism.
Thanks a lot, Paul. Yeah, good to see you again.
You are a herpetologist, amongst other things.
I am, yeah.
What is a herpetologist?
So a herpetologist is a scientist that studies reptiles and amphibians.
And a lot of those reptiles and amphibians, we'd call them conformers, is that right?
That's true, yeah. Generally, I think like as you move up through the scientific ranks, you tend to refer to them more as like ectotherms rather than conformers, but yeah, it's basically the same thing. Unlike us, like mammals and birds, the homeotherms, they're unable to control their inner body temperature. So they have to use sort of behavioral mechanisms like basking, things like that to maintain their like thermal optima. And at those sort of temperatures is where things like digestion and like metabolism, movement, and all of these sort of bodily processes can occur.
Sort of reptiles and amphibians, because they're not regulating their internal body temperature, they'd have quite low metabolic costs then involved in that.
Yeah, that's exactly it. So you've probably seen examples on TV of like these snakes eating a massive meal and then that like suits them for the whole rest of the year. That's because of, like you say, their metabolic demands are much lower than the mammals because they don't need to spend so much energy keeping themselves warm. They can just go and sit in the sun for a bit. Some reptiles, and I'm actually not sure about amphibians, but they can actually see UV. They've got an extra cone cell in their eyes. So basically that will then identify to them where the best basking sites are in their environment. So they'll move over to that and they'll just sit there. There's some sort of behaviors that they can do where they like flatten out. I don't know if you've ever seen a lizard basking in the UK, but they'll often look like they've kind of been stepped on. They like really flatten themselves out, squish their ribs out, maximize the surface area for the sun to be in contact. And that will help to warm them up quickly.
And that then allows, I guess, their metabolic rate to increase and for them really to get on with their day, I guess.
Exactly that, yeah. And that you'll get these really distinct patterns of activity with the reptiles. And this happens throughout the world, not just in temperate climates or tropical climates, where in the morning, like just when the sun rises, there'll be a big peak in like basking activity where they're trying to warm themselves up ready for the day. Once they've reached that thermal optimum, they'll then head off and start foraging, and the body temperature will be slowly declining. So they maybe will kind of top it up on the way in patches of sun. But generally, like right in the middle of the day, especially in places like Australia or the Middle East or something, it's just way too hot to actually just be sitting under the sun. You'll just cook. So quite often in like the very center of the day, they'll be hiding away somewhere or resting. And then in the late afternoon, again, they'll come out and bask to kind of help with metabolism, digestion of food, ready for the night, and then to start it all over again the next day.
And I guess that kind of reduces the number of ecological niches that are available to them then?
Yeah, well, especially when it comes to like geographic locations. So obviously, like the further north you go, the colder it's going to get. And with that, you see the diversity of reptiles and amphibians reducing significantly. And yeah, like equally within environments, they need to have a few more things in place than the mammals. It's not quite as easy for them to just get on with it.
And we're fairly far north, certainly compared to the tropics here in Scotland and in Glasgow. Am I right in thinking we've got about 6 amphibians and 3 native reptiles? What can you tell us about them?
Yeah. So I think in Scotland, like you say, you've got 9 native species. In the UK as a whole, there's 13, but that's just because we have a few species which have like really, really restricted ranges, and especially in like southern England, like something like the smooth snake. But yeah, up in Scotland, your reptiles, I believe you've got adders, which is the UK's only venomous snake, slow worms, potentially, I think common lizards you get up there. And then for the amphibians, toads and frogs. Potentially some species of newts.
Not many people might be aware, obviously, that we've got a venomous snake in Scotland. What can you tell us about the adder?
So the adder is actually a really, really cool species. They're the furthest north ranging species of snake in the world. So in places in Scandinavia, they actually range within the Arctic Circle, which for a conformer is like pretty cool. Some sort of adaptations that they've got to that to overcome it are things like giving birth to live young. Because if you just go and lay an egg somewhere that's with like the wildly varying sort of weather patterns that we have here in the UK, they're not going to do too well. So like maintaining the young within the body and then going out and basking and trying to keep the temperature at a sort of semi-stable state, a much better way of developing the young.
But for any reptile and amphibian, the sort of climate in Scotland must be a bit of a challenge.
Reptiles and amphibians that we have in the UK are like, they're not like the biggest, brightest and the most spectacular species on Earth. But they are very, very cool because they're like super hardy, have various adaptations that can help them with surviving the cold, basically. Because, well, especially up in Scotland, you guys get some pretty cold winters.
Yeah, we're pretty hardy.
And so species like the common lizard, they can actually survive freezing. So there was one experiment I was reading about in a paper, which like I think it was in like the early 2000s, and for that sort of time frame, it's kind of like ethically dubious, but they were basically just freezing lizards and seeing after how long they could thaw them out and they would survive. I think something like at 50% of the body being completely frozen, after 24 hours, these lizards could be thawed out and then come back to life.
Wow.
So yeah, they do that by basically filling their cells full of glucose and sugar. And what that does is it stops the ice crystals just like exploding the cells, basically.
Definitely a little bit ethically dubious if you're a lizard that sticks you in the freezer for 24 hours. Yeah. Ollie, we first met in the Sani region in the Amazon in Ecuador back in 2018, maybe, I think it was, that we first met up. And you've obviously spent quite a bit of time out there since. What can you tell us about the work that you do out there and about the neotropics in general?
So I think when in 2018, that was my second year out there. I did one more season the year after you guys, and then obviously everything fell apart in 2020, as we all know about. Since then, I went back to Ecuador. I've been back 2 times, which were late last year and early this year. And I've sort of literally branched out. And I've started working in the canopy in the jungle there. So been running some expeditions to climb into big trees in the forest and look for new species. And so far, I found a new species of frog and a new species of orchid.
And did you get to name the species?
So I would have done had I applied for the correct permits that would have allowed me to take samples, which I didn't do. So I just had to wave my brand new frog species goodbye as I let it go back into the canopy.
What would you have called it?
My team that I was out with had some interesting ideas, ranging from Beyonce's tree frog to all sorts of other crazy things. But I probably would have named it after something about the canopy. I think it was the place where we found it.
Remote scientific expeditions in the rainforest can be incredibly rewarding, allowing researchers like Ollie to discover new species, study unique ecosystems, and contribute to our understanding of the natural world. However, these expeditions also come with significant risks, including exposures to extreme weather, dangerous wildlife, and potentially life-threatening illnesses and accidents. Ollie went on to share a couple of the more uncomfortable moments of his time in the rainforest.
I can only really think of one hairy moment that actually involves a conformer. The rest of the time, it's normally with people. Like one time, it might have even been 2018. I was cutting a trail with one of the guides in like a really heavy downpour of rain, and as he swung back with his machete to cut a big branch, it slipped out of his hand and it like flipped through the air and hit me straight in the chest. But luckily, with the hand length, otherwise that would have been pretty nasty.
Yeah, a long way from help there.
Yeah. In terms of conformer accidents, I got a bit too overconfident with a caiman once and it bit me in the thumb. So that was exclusively my own fault.
And you still have said thumb?
I do, yeah. I got away very lightly on that occasion. But it was a good reminder that nature will always win.
You've done a lot of work in a lot of remote and exciting places, and you've mentioned that the people are obviously the biggest threat. What sort of damage are people doing to these places where we see such diverse range of species, particularly in terms of amphibians and reptiles?
It's quite a complicated issue, really, and it's like a real sort of mixture of like social, economic, and various other factors. Obviously, the places on Earth with the most diversity of these conformers, the reptiles and amphibians, are the tropics. So, well, just as an example, the 4 out of the top 5 countries for amphibian diversity are all in South America. These places are in developing countries, so like the economy is growing and populations are growing, and as a result of that, more land use is required. And on top of all that, you've got pressures from the Western world as well for things like soy and all of these issues that we hear about all the time in the news. Well, I mean, to cut a long story short, there's a lot of habitat destruction going on, and that's probably the leading cause of declines for reptile and amphibian species. Another massive factor at the moment is this amphibian chytrid fungus, which some listeners may be familiar with, but it's basically this fungal infection that's spreading throughout most of the world, where it causes amphibian skin to thicken to the point at which they can't pass ions or like different molecules and stuff through the skin, and basically they can't breathe and they'll die. And that is causing like major extinctions, especially in Central and South America, of amphibian species. And I mean, there's a lot of research going into it. Yeah, things aren't looking great currently.
It's a bit topical. It's got a touch of The Last of Us about it.
Yeah, very much so. It's based on the cordyceps fungus, which is like the ant zombie fungus, isn't it?
Yeah.
If the writers have been true to the fungal world, it will be spread by a sort of spore, which is the same thing with the chytrid fungus. They have these little spores that they kind of look like sperm cells with a little tail, and they can swim about and they live in water and transfer between frogs that way. And they bind to any parts of the animal that has keratin. So that's the sort of protein that makes up your nails, hair, and skin. So on a tadpole, for example, that's like their mouth parts. So when you're testing for chytrid, you need to like do a little swab of the mouth parts. If you're testing an adult frog or something, you swab all over their legs and their back. You can pick up any fungal spores from that. So an amphibian is just a very different living being to a mammal. And so a lot of their gaseous exchange and, like I was saying, like transport of ions, which controls a lot of our like cellular processes. So things like calcium ions that enable messages to be passed across synapses in neurons. All of these different molecules and ions, although they're like tiny atoms, and they're controlling these processes in the cells, which then control the animal's movement, for example. So everything's sort of linked together, and something as simple as just having your skin being a bit thicker can block one of those pathways, which then leads ultimately to asphyxiation of the animal.
And that's something that you said traveling around the world then? And
yeah,
that's happening now?
That's happening right now. The leading theory for the spread of chytrid for ages and ages was the African clawed frog. It's called Xenopus laevis is the species name. You've probably seen one before in a pet shop. They're like a white, sort of almost 2D frog with like kind of fingery claw things, quite common as a pet. But at some point in the 19th century, I can't remember the decade exactly, someone found out through some, in my opinion, quite weird experiments that you can they're basically quite an accurate pregnancy test. So if you take a wee on the frog and then it starts producing eggs, then like you're pregnant basically. Obviously not you, Paul, but
No, I doubt it. Might look pregnant sometimes, but
So these frogs were like shipped all over the world as these sort of make-do pregnancy tests, and they are also asymptomatic to chytrid infections. So for ages and ages, that was the leading theory. But I think there was another paper that came out. I want to say in like the late 2018 or something like that. That was basically saying that actually they think now that the chytrid fungus originated in Korea, and through various, mostly human-mediated means, it's been spread all over the world, and especially in the pet trade. That's like a big factor in it. People like getting a pet frog in somewhere because there's a lot. Although it's getting much better in captive husbandry for reptiles and amphibians now, there used to be a lot of like wild catching of animals for the pet trade. So people would get these animals, and then the spores of they're infected with the fungus. The spores then go into the water. When you're cleaning out the tank or something, you shut the water down the drain, and it goes out into the environment. And although the spores don't live forever, they're quite hardy, and they can live for a certain amount of hours. And in that time, it's enough to infect native populations, and that's basically how it's spread across the globe.
What advice, Ollie, would you have for young people who are still at school and might be interested in herpetology, or maybe even finding a little bit more about it?
I think if you want to get some more exposure to any herbs, then just maybe join like your local amphibian and reptile group. They're always after volunteers to help with surveys. It's like quite important, sort of, especially in the UK. A lot of our like wild spaces are getting pretty battered. So if you can go out and help look for any species that are of like importance, like great crested newts or any of them, to be fair, it's always nice to go and see them in the wild. And then also I'd say there are many different ways of getting into herpetology, and I think it's important to recognize that like you don't have to be a scientist to help out with it, or even within science there's like various different disciplines where you can still interact with these animals and have, like, for example, if you work with disease, or if you're interested in diseases, you could go and work on the chytrid fungus and actually have like a massively helpful impact on these animals. Yeah, you don't need to go like directly down a herpetology route or even a biology route to end up working with these animals or helping them out.
As Ollie has said, there isn't always one straight path to get involved in a specific area such as herpetology. The biological sciences are a vast and complex field, encompassing the study of living organisms from the tiniest microbes to the largest mammals. It's a rapidly evolving field that's constantly revealing new insights into the complexities of life on Earth. For anyone that's interested in pursuing any career linked to biology, it's essential to have a strong foundation in the broad concepts and principles that underpin the biological sciences, something which Professor Whelan also believes in.
And certainly my advice to anyone who's interested in doing work in the biology world is not to become a specialist too early on. I've had people that I've interviewed for jobs in their late 40s, early 50s, that basically have put off life with a view to actually getting this big research job. And they ended up so specialized in terms of the areas that they were working on that in reality, they really could not find a niche for themselves. And it was a bit shocking and very disappointing to see that happen with people that had huge amount of ability, but really hadn't got the broad practical skills that are needed, certainly in the fisheries world. It's not just a matter of knowing biology. You really have to know about the other creatures and the other life forms that are around the actual creature that you might be looking at the time. And also all the lovely practical skills in terms of being able to use boats and engines, being able to use the various forms of equipment. And then through the geography, I've always been very interested in mapping. Again, once I got my hands on GIS, on geographical information systems, the idea that I could actually physically map in such detail the areas that I was interested in, and then overlay on that map all of the different habitat types and so on. that was just mind-blowing. And the whole thing, just in my mind, it's all the one picture. It's not this very, very finely focused area. It really is truly the ecology of the system that I'm looking at, that I'm interested in.
And within our higher biology course, can we look to compare marine and estuarine invertebrates and the response to the variation in salinity? And of course, that very much fits into that blend of experience that you've had.
Yeah, I think the salmon and its close relative, the sea trout, I generally tend to not, in my mind, separate out the two things really, because they are just so closely related. And in many ways, we might chat about it a little later on, many ways the sea trout are even more interesting than salmon. But both of those creatures have this extraordinary ability to be able to move from fresh to salt water. And the term we use is anadromous. And anadromous are the creatures that basically feed in salt water and reproduce in fresh water. But the whole group we call the diadromous fish. So that would include eels. And eels do the reverse, which we still don't know why. Why would you come out of a very, very rich ocean to spend up to 50 years in fresh water and then go back to the Sargasso Sea to lay your eggs? It doesn't seem to make sense, but that's exactly what they do. But these creatures that make this transition between fresh and salt, really, as you were saying, the physiology of this is absolutely fascinating. So if a fish is in freshwater, if a salmon is in freshwater, it has to avoid drowning because it's surrounded by all of this fresh water. So it has to keep all of the salt inside its body. But yet when it makes the transition to the salty estuary, and particularly to the full salt of the ocean, it still has to take in water, but it has to take in water that has a lot of salt in it. So it has to have a system whereby it can actually rid itself of the salt. And it has to be very careful not to dehydrate, because you have a very, very dense body of water surrounding you in the ocean. You have a high salinity pool of water constantly surrounding you. So the way they do that is they have these fantastic mechanisms in their gills, in their gut, and in their kidneys, so that they can constantly pump salt and stay very comfortable in that salt environment. But the fact that they can then reverse that, having gone to sea and fed for a year, 2 years, even sometimes 3 or 4 years, and come back from the marine and then switch again in the estuary and go back up freshwater is really extraordinary. And the sea trout in particular do this absolutely constantly. They move from fresh to salt, salt to fresh, fresh to salt, and they do this at their ease. They really astound me in terms of their ability to be able to do this. And certainly it makes them very special fish, but it also, as you can imagine, it puts them in a position where they are really at extra risk because of this. And again, the challenges that they face in terms of the long distance migrations that they have to undertake, the fact that they have to switch their diet from a freshwater based diet to a sea-based diet. They have to move as quickly as they can away from little planktonic forms of organisms in the surface layers of the ocean. They move away from that as quickly as they can, and they feed on tiny fish. And once they start to feed on fish, then they can make the growth that they need to make in order to be able to produce eggs and sperm and come back to fresh water to reproduce. But the journey they make and the synchronicity that's involved, where the fish have to migrate at a point in the cycle of the ocean where they have the food available to them that's of the right size and the right nutritional content. So overall, it is extraordinary what they achieve and seem to do it in a sort of really effortless way.
And of course, although it might appear effortless, that kind of regulation of their internal environment requires energy to achieve that. So I guess their metabolic costs have increased.
Yes, absolutely. Let me just tell you a little anecdote, because I think it was just so profound. It really hit me right between the eyes when it happened. So I mentioned earlier that I was a director in the Marine Institute, and we had a cooperation going with a big organization in the US called NOAA, NOAA. NOAA look after the seas and so on in the US. And as part of this cooperation, we had an exchange whereby the directors from the Marine Institute went over for 2 weeks and were actually paired with one of the directors from the ocean part of NOAA. And I was paired with an oceanographer, somebody who knows all about charting oceans and the currents and everything else. And I was a little intimidated, to be honest, because this guy was super famous, and I was really lucky to spend a couple of days with him. So I sat down in front of this guy, and I was a little bit gobsmacked and a little bit nervous of him. And he put me at my ease immediately. He said, well, tell me about what you do, So I told him then, and he knew nothing at all about salmon. And he stopped me when I was telling him about the life cycle of the salmon. He said, let me start that again. He said, so you're telling me that these creatures go to sea at about 12 centimeters? And I said, yeah. And they come back after 14 or 15 months, and they're 55 to 60 centimeters. And I said, yes. And he just looked at me, and very simply and very cleverly, he just said, they ain't doing much swimming. So the energetics that are involved in this is absolutely fascinating. So these little creatures who know nothing about the ocean have to go out there in May, go to the surface layers of the ocean, find the right creatures to feed on, and they have to utilize the currents. There's absolutely no way they would be able to swim from here to the east coast of Norway. We know exactly where they go now, a place called the Vorning Plateau. So to get to the Vorning Plateau, they just couldn't burn up energy all that way. So I imagine them inside these massive great currents, and they are moving, but they're moving left and right, if you like. They're actually using and hitching a ride on these big motorways and feeding voraciously. But the food has to come to them in the right quantities and at the right time for them to actually do no swimming, for them to overcome that energy deficit that would be involved if they try to do this without the support of the currents and without the support of the very rich feeding that's in the oceans. But that was an incredible statement, that statement, they ain't doing much swimming. I'd never thought of this in terms of energetics. I've never forgotten that. And it has been a really important comment that I really have been very grateful to that guy for down the years, because it does make me think about their journey in those terms.
As regulators, they obviously maintain that internal environment regardless of the external environment. And I guess by using metabolism to control that internal environment, that really increases the range of possible ecological niches that they can exploit.
Oh, yeah. And they are, as far as we know, they are absolute wizards at using currents. And that's the way a lot of this regulation, we think, probably takes place. Now, I should add that, remember, we're only at sea looking at these creatures since 2008. This is ridiculous, but we were over 100 years looking at salmon based on what was coming back into estuaries and back into our rivers. But in 2008, then we put together this big international program called Salmon at Sea. And we went out on boats and we actually followed them in terms of finding out where they went and trying to find out then much more about how they actually did this. But certainly what we have found is that up to about 750 grams, about a pound and a half, these fish are more or less, we feel from the energetics point of view, maybe trapped is the wrong word, but certainly they would much favor being in areas where they're not expending much energy. But then it would seem that about 750 grams early in the first winter that they're at sea, then they truly become masters of their own environment. They then start to roam around in the surface layers of the ocean. And we know that they can go far, far north into the Arctic. They also move across, if they're not coming back after 1 year at sea, they move across the top of Iceland, through what's called the Irminger Sea, and around the southern tip of Greenland. And they feed for 1, 2, or 3 years more in the rich waters around the south of Greenland. So these fish have this amazing ability to be able to regulate their energy balance, both internally and indeed, not that they're regulating the external factors, but using those external factors to help them to optimize the food resources that they're finding so that they can actually mature and come back in the very best condition and with the very height of nutrients in their bodies, because they don't feed at all when they come back into fresh water.
And I guess there must be some pretty specialized techniques when it comes to studying those long distance journeys.
Yeah, well, I was really, really fortunate this year. I'm one of the few people on the planet that's actually managed to catch Atlantic salmon feeding in the ocean. And we did that in order to put very sophisticated satellite tags on them. So my friend John Carr started this program about 4 or 5 years ago in some of the fjords in Greenland, where these salmon who are going to stay at sea for several years, where they feed and where they hang out over the extra years, if you like, that they spend in the ocean. And John managed to actually find locations where the fish could be caught while feeding on small fish with the same techniques you would use in the loch in Scotland, trolling and so on. So we spent some time in Greenland together this year. He'd been there several years before. I think he's done 3 years or 4 years there now. But we had, between us all, we had about 150 salmon that we were able to tag. And these satellite tags, they're like very much like a big cigar that sits on the back of the fish. It's exactly the same technology that's used on whales and dolphins and seals. And they're programmed so that after a certain period of time, they actually, there's a chemical reaction and they then split away from the back of the fish. They float to the surface, and the minute they hit the surface, they transmit their data then to a satellite. And as we sit here today in early March, some of those tags are programmed to now start actually leaving the fish from this week onwards. And over the next 3 to 4 weeks, those tags will be signing in to John's computer in New Brunswick, and he'll be sending me some of the data that's coming off the fish that we tagged in October. So this is just mind-blowing technology that's available. And we're very lucky in Scotland in that we have, through funding that we've received to the Atlantic Salmon Trust from Marine Scotland Science and in partnership with Fisheries Management Scotland, we have a whole series of experiments off the west coast of Scotland now, where we have tracking arrays in place to follow the movements of the smolts, as the young salmon as they move out, and hopefully increasingly we'll have the ability then to monitor the adult fish as they come back. But that's all down to this very, very modern technology that's now available.
Wow, that sounds fascinating. And do you have any expectations of what might come back in the data?
Certainly in terms of the adult fish, we don't have any great expectation, to be honest. But based on the material that they've got over the last number of years, it is beginning to follow a pattern. And certainly, which is very good for Scotland and Ireland, most of the fish in the particular area where John is tagging, most of the fish that are from Europe are either Scottish or Irish fish, predominantly Scottish and Irish fish. About 50% are Scottish and Irish fish. The rest are from North America, primarily Canadian. So that gives us great hope that some 50% of the tags that we put in, that they will start to pop off, and we will have, from the genetics, we'll have a very good idea what part of Scotland or Ireland these fish came from. And we'll begin to build a picture about their migration path backwards, having already built a picture in terms of the smolt migration during the initial years.
It sounds like there's a significant number then of salmon around Scotland and Ireland.
Well, let me give you the overall numbers first, and then we can talk a little bit maybe about Scotland and Ireland. So, tragically, we have seen a very big drop in the overall population. So we were in a situation in the 60s and 70s when we had somewhere between 8 and 10 million Atlantic salmon in the ocean, and that would be Atlantic salmon from everywhere, from North America and also from the European side. And that has plummeted to a situation now where we reckon there's probably 3 million or less fish at sea. So proportionately, the number of fish coming back would have greatly reduced in that time. So I don't have a figure in my head for Scotland in terms of the numbers of fish that are coming back, but it would certainly be, again, it's probably about a third of what originally would have been there. It's complicated in Scotland in that we're very, very fortunate in Scotland to have a high proportion of bigger salmon, what we call multi-sea winter salmon. The salmon stay at sea for more than 1 year. Whereas in contrast, for example, in Ireland and Newfoundland, predominantly it's much bigger numbers of salmon, but they're much smaller. So the numbers game is really important, but equally important are the number and quality of eggs that are laid. So if you have a situation as you have in Scotland, where you have these beautiful, big, older salmon. You have big, very, very nutritious eggs that these fish lay. So that's a fantastic buffer against the problems that they're facing into at this stage. But there are certainly in some rivers really serious concerns. And the Scottish Government has already publicly announced on several occasions that Atlantic salmon, tragically, are in crisis. And we really need to be doing something to make sure that we do the most we can to try and boost the number of juvenile fish that are going to sea. That's really the main focus at the moment, is to try and get really high quality salmon smolts that are in as high a number as we can back into the ocean to see can we counter some of the problems that we know are now taking place in the ocean.
And would I be right in thinking one of those locations is on the Moray Firth?
So in terms of the Moray Firth, then, what we did was we chose 7 rivers in the Moray Firth, big famous rivers like the Ness and the Devon and so on. And the Moray Firth is very special in the sense that those 7 rivers represent about 20% of all Atlantic salmon in the whole of the UK. So really important rivers. So the main purpose for the project was twofold. First of all, to try and look at what were the mortality rates like in freshwater. What were fish facing on their way down to sea as little smolts? And this was really important to get this baseline. A, we needed to know what the situation was at the present. And B, we need to know what's the potential. How can we increase those numbers and the quality of the fish? And the second objective then was to look at the first 100 kilometers out to sea. This seemed an impossible task. I mean, 60 miles out to sea for people who are fundamentally freshwater biologists is really a daunting task. But the staff that were involved, it was extraordinary what they succeeded in doing. So what they had to do was lay these receivers along the bed of the sea out for about 100 kilometers out to sea. And as the fish came downriver, there was receivers in the rivers as well, receivers in the estuaries. They basically clocked in. They basically went by the little bus stop, clocked in, then to the next one. And we could see then the mortality rates. We could see the movement of the fish. We could see any strange movements of the fish. And interestingly, we did manage to actually track these fish out that distance out to sea, which was really a fantastic success. And it means now that we can get to the second part of the program, which really is to look at what's causing problems in freshwater. What are the factors that, particularly the man-made factors, that we could eliminate or we could reduce in order to try and boost the numbers of the smolts and the quality of the smolts going to sea, and also to make sure then that we continue to monitor our success and to be able to quantify if we really are making a difference in terms of increasing the actual outputs from these rivers.
In one of our previous episodes, we had Professor Neil Gemmell from New Zealand, who travelled over to Scotland to search for the Loch Ness Monster using eDNA techniques. Am I right in thinking that's something that you guys have used as well in terms of identifying some of those pinch points along the smolt's journey?
Yes, we have indeed. And I mean, there's a couple of some of our detractors maybe might call them obvious, but in reality, you have to have numbers. As scientists, we have to have numbers and show that these really are problems. Even though intuitively you might think they were problems, it's great to have numbers to show that they are problems. So we certainly have really good numbers now showing that these pinch points, any sort of man-made factor that actually delays these smolts, that puts any sort of obstacle in the way of the smolts, that's a big no-no. There's no doubt about that. And if you were a predator, where would you go? You'd go to those pinch points and you'd wait for the water levels to drop or the fish to be confined in one area. And you'd have a heyday. You'd have a great time then in those particular locations. The other thing that's very obvious is just how critically important correct water levels are. And the water levels have to come in the right volume and at the right time. And looking at what we're facing into in terms of climate change, it's really important that we're conscious of this, because in rivers where we have the ability to regulate flows, there's a lot of hydro, as you know, in Scotland. In the past, that would have been seen as a big negative. There may actually be a positive to it in terms of climate change. Maybe we can now in a much better way, regulate and artificially compensate for some of these very low water conditions that we have seen in the past. But in terms of the predators themselves, we had been working with colleagues in the University College Dublin, just down the road from where I am here today. We had been working with colleagues there since about 2016, using this technique of environmental DNA. We were looking at the time at bycatch. We have this problem when the salmon smolts are going to sea. They feed on food that's very similar to the food that mackerel will be feeding on. They get mixed up in the huge big mackerel shoals in the North Atlantic, and we were concerned that they would be caught. And we know they are caught, but we don't know what level of catch is there. They're caught as a bycatch with all of these mackerel, when the mackerel are being caught on the surface layers of the ocean. So my colleagues then developed techniques so we could take samples in the holds of some of these boats, and we could actually detect salmon DNA in the holds of the boats and get some idea of the quantity of DNA that was present. So because we had that background in the environmental DNA area, or my colleagues had that background, we then suggested to them, would it be possible to actually start monitoring some of the predators? So for the past number of years, we've been doing an absolutely disgusting job. We have been going around collecting bird poo, collecting bird scat from a range of different predators. And this year we extended that to otters as well. And those samples are currently being analysed. So what we're able to tell, the food that the fish are feeding on at any particular time. And hopefully we might be able to see a switch. If they do at these pinch points switch to smolts, hopefully we can pick that up in the scat, because we designed the program so that we took samples before, during, and after the smolt run. And as I say, those samples are just being analysed at the moment, and we're really excited to see the results.
Why are our wild salmon stocks so valuable?
Well, if we have another 4 or 5 hours, I might be able to explain that to you. But I think in reality, from an ecological point of view, they really are they really are the top of the tree. It's not so much that they're sort of the top carnivore, but that the top ecological species, in the sense to try and put together all of the threads that we've been speaking about, and to make sure that they all coalesce in the right way. It means that the rest of the ecosystem has to really be at its very best to produce the quality of smolts, the number of smolts, the genetically fine-tuned populations of smolts that are going to be able to make these journeys and return. So if we basically can get the salmon right, we can get the rivers right, and everything else that surrounds the salmon will be in order, will be as God intended it to be. That's certainly our view. So making sure that these keystone species are maintained is really saying, no, it's not so much the species. These are the indicator. These are the aquatic canary, if you like, in the aquatic goldmine. These are the species we must absolutely conserve and enhance if we're to try and protect the overall environment. And the great thing now is that people are beginning to understand that this is not a freshwater fish. This is not an estuarine fish. You must look at the totality of the salmon's life to understand how you can protect it. And by doing that, you're protecting all of those different zones that the fish is moving through. So that's why I think it's a really important keystone species.
What would the overview of that salmon's life cycle be?
Yeah, so there's only 2 dates you have to remember. Christmas Day and my birthday. They're the only 2 dates that you have to remember. And so what happens is that in the autumn time, September, October time, the fish then start to turn red. They've come in from the ocean. They haven't been feeding since they were at sea. And they move up then looking for areas of very fresh, very, very well aerated gravels. Gravel that has lots of water percolating through it, that has lots and lots of oxygen. And they lay their eggs then in the gravel. And the peak time for egg laying in Scotland would be in or around Christmas Day. So Christmas Day is a really important date. You get lots of pairing going on from maybe late November onwards, but it tends to be around Christmas time that you get the peak of the eggs being laid. And some eggs will be laid then maybe a little bit into January, but generally it's that Christmas period or a little bit before. The eggs then are in the gravel, but when the female is laying the eggs into the gravel and making what we call a red or egg nest, she uses her pectoral fins, her front fins, to test for the draw of the water. So she will only lay the eggs where there's a really good flow of water going through the gravel. Because if she was to put the eggs into the gravel where there was no oxygen, those eggs would not survive. So if you can imagine these little gaps between the little particles of gravel and stone, the water's percolating and bubbling down through that. The eggs are as happy as Larry down in that particular location. They get little eyes very quickly, surprisingly quickly. There's two little black eyes looking out at you. And as they develop then, they tend to develop front and rear. So the head then pops out from the egg itself, and the tail grows out from the other end. And the egg itself then is the tummy of the fish. And it's just like a chicken's egg in the sense that it's full of yolk, it's full of nutrition. So they're not feeding at all when they're in the gravel. And they absorb in this yolk sac, as it's called, into the tummy. And they use that to feed away until around my birthday, which is the 1st of April. And around that time, sometime around mid-March to mid-April, but generally around the 1st of April, they'll actually come out and they will sit on top of the gravel. And they will start to then feed naturally and feed actively. And they'll stay in the river. Generally in Scotland, they will stay in the river for 2 or 3 years. And then after that, they go to sea then as these little silverfish we call the smolts. After they reach the ocean, they will stay in the ocean for 1, 2, or 3 years at sea. And in Scotland, the bulk of the fish originally were these big 2, 3 sea winter fish. And in the smaller rivers, then you get a lot of the one sea winter fish, as we call them, the fish that have spent 1 year at sea. They'll come up and they lay their eggs, and then the cycle starts again. And surprisingly, quite a number of salmon actually survive spawning. And we tend to imagine that it's the same as the Pacific salmon, where all the salmon die after spawning. No, there's quite a good number of fish that actually survive spawning. But once they get back to sea, they find it very difficult. And only about 5 to 7% of the fish that go back to sea return to spawn a second time in the Atlantic salmon. In the Pacific salmon species, those fish just don't survive at all. That's the way the system is. They have one shot at it, lay the eggs, and then they die. And the nutrients from the fish are hugely important in terms of fertilizing the rivers where the eggs are.
What work's been done in genomic sequencing of the salmon, and what have we maybe learned from that?
Yeah, because of the aquaculture industry, really, we have now got really detailed knowledge of the salmon's genome. And certainly because of the new technologies that we have available now, we have certainly with using the older microsatellites and now using SNPs, we have very clear maps, if you like, of the different populations, certainly the key populations all around the whole of the North Atlantic. And what managers now use as a standard technique in looking at the actual distribution of the populations around Scotland, that's all relatively new. It would have been only over the last 20 to 30 years that those techniques were developed. And from this big program, the SALSI program, we originally developed a key set of genetic markers, and they have been refined now. And there's a very sophisticated set of markers that can actually define the regions that the fish have come from when you're sampling for them in the ocean. So that's a hugely important move forward. And I have no doubt that in the near future, we'll have even more refined techniques that will be able to tell us on an individual river basis where these individual river fish are moving at sea and how they are moving at sea. And the work that I mentioned in terms of the environmental DNA, if we can collect enough DNA at sea, imagine a situation where not alone could we identify the presence of salmon, but we could identify some of these regional markers at sea. Then we would really be in a situation where we could start to delineate these corridors at sea and to manage them in a very proactive way.
And you mentioned aquaculture there, Ken. Am I right in thinking that would involve fish farms?
Yes. So you have a situation now where, as we sit here in cages around the world, there's about 700 million salmon being reared in cages all over the world, from a situation where certainly in the early 1980s, this was only just the very beginning of this big aquaculture industry. It has really, really bloomed since then and blossomed.
With those numbers, they're being intensively farmed to support the production of food for ourselves. And there's quite a bit of that taking place in Scotland as well. How does that practice impact the wild salmon, and what impact do the fish farms have on the environment?
There are two really big issues that we have in terms of the aquaculture industry, and it's an absolutely tragic story. Whether it's Scotland, whether it's Ireland, whether it's Norway, in all of these countries, the development of the aquaculture industry was actually encouraged and allowed to move far, far faster than the science. And even when we were sounding alarm bells, the pushback was extraordinary. It was absolutely extraordinary. And from a personal point of view, I can attest to this. I mean, we had just such a rough time as scientists trying to explain to people that we were actually trying to produce, real science that indicated very clearly that there were problems and they needed to be tackled. And we were told constantly, go back to the drawing board, go back to the drawing board. And even when we had large amounts of data published in peer-reviewed journals, the management action still wasn't taken. So what management action were we looking for? Well, first of all, the really big issue that emerged firstly was the growth in terms of what we call sea lice. So these little sea lice are quite natural. They're on wild salmon and wild sea trout. But they're specific to salmon and sea trout. They can't feed and grow on any other creature except salmonids. But if you have a situation the same as you would in a pig farm, where you have a huge number of bodies, well, you're going to exaggerate the number of parasites that you're going to have. And certainly the sea lice issues that we had by the early 90s were extraordinary, the levels of lice that we had on the fish farms. And we know solidly there's really good science in relation to sea trout. We know the consequences of that on sea trout. It led to massive mortalities of sea trout in certain locations. The salmon evidence is growing stronger and stronger all the time. The salmon situation is very different because the fish are passing through relatively quickly. They're going out to sea and you don't really know what happens. Whereas in many bays, the sea trout stay in the bays and you can monitor, as has been done over the last 30 years, you can monitor exactly what's happening. And the tragic thing is, because I had personal experience of being involved where I ran a team that was involved with research and development for the aquaculture industry for a wide range of species, including salmon, as well as being responsible for wild fish. And we did manage over a few years to get to a situation where we found a balance between the aquaculture production and the wild fish protection. But it was at a level that nowadays would not be commercially viable. And that's our problem. Our problem is that I always look on it as each bay having very, very much restricted capital, natural capital within that bay, in terms of the bed, in terms of the food resources that are in the bay itself, in terms of its ability to deal with any new and very intense farming operation in that bay. How is that affecting the overall sustainability of that particular bay and its capacity to actually to be able to host that amount of extra bodies? I think we have totally and completely overtaxed the ability of our bays to actually deal with this influx, this massive influx of additional fish flesh, both in terms of the quantities of food that are pumped into the bay and the presence of the fish themselves, the release of lice. But then there's another more insidious problem, and there's a fantastic report available in Scotland that was produced by a friend of mine called John Gilby a couple of years ago. And again, going back to genetics, because the genetics of these farm fish is quite different to the genetics of the wild fish. And John was able to show where the rivers were suffering as a result of crossbreeding between the farmed fish and the wild fish. And consistently, the problems were in the areas where the farms were located. And I have no doubt, based on my own experience, that a balance could be found between the fish farming production side of things and the wild fish. But I'm afraid, as I say, that the fish farming industry would have to seriously lower its expectations in terms of the quantities of fish that it can grow in any particular bay. And that to me is the crunch point.
And that crossbreeding, I assume that's from the farm fish escaping.
That's the farm fish escaping and then crossbreeding with the wild fish in freshwater. But a lot of it, remember, people always think of that in terms of the big fish escaping from the ocean. But there seems to be a really serious problem in terms of baby salmon escaping from some of the cage systems that you have in the lochs in Scotland. And because of the fact that they acclimatise to that particular watercourse, they tend to behave very much like wild salmon in terms of their migration patterns and their movement back into fresh water. Whereas if fish escapes from a cage, it's been artificially fed. It's certainly not an environment where it's comfortable. It's instinctively running up into fresh water. Its success rates are going to be quite low. Whereas any of the baby fish that escape in freshwater, any of them that actually survive, they're going to mimic very closely the behavior and I would imagine the survival pattern of the wild fish.
For anybody that's interested maybe in seeing some salmon, Ken, where would you suggest are the best places in Scotland to see salmon, and whenabouts would you recommend they go and try and see them?
Well, again, the really ideal time to go and see them would be around late September into October. And there's quite a number of different falls around Scotland where it's possible to actually see salmon. I know that my colleagues in Fisheries Management Scotland are just a little bit anxious about this. They don't want to be in a situation where very large numbers of people congregate with relatively small numbers of wild fish about in the rivers, and they would be disturbed, particularly if they were spawning. So the best advice I can give is that if people contact Fisheries Management Scotland, they'll be able to get advice in terms of guided locations where they can actually go and see salmon spawning or indeed see salmon jumping waterfalls and so on. And I think that's the best advice, because otherwise, if I was to mention one or two particular locations, I'd be quite fearful that it might actually lead to a situation where very large numbers of people, relatively speaking, appeared at a very sensitive time for the fish.
That's the assuming we've got big numbers listening to the Higher Biology podcast, although it has been listened to in over 100 countries, so maybe it would spark some sort of tourist.
You might get your own migration back to the salmon spawning rivers. That'd be fantastic. But there is just so many people now. As a colleague of mine that works with us in the AST, who's actually doing quite a bit of stuff now for Spring Watch and for Autumn Watch and so on. And Chris Conroy is just a wizard with his cameras and his GoPros. And there's just so many wonderful clips and video clips and everything else available now of salmon jumping and salmon spawning. That's again a very, very good substitute for people who are interested in seeing what's happening.
Absolutely. And the last thing, really, Professor, is just to say a huge thank you for joining us on the Higher Biology podcast and contributing in our work around so many aspects of the course. Thank you very much, Professor Whelan.
Not at all. My pleasure. Thank you very much indeed, Paul.
Many thanks again to Professor Whelan for his input in this episode. Wild Atlantic salmon are a remarkable species that regulate their internal environment through a variety of physiological and behavioural mechanisms. These fish are able to maintain their internal body temperature and fluid balance despite changing external conditions by controlling their metabolic rate and adjusting their behaviour in response to environmental cues. One crucial mechanism used by salmon to regulate their internal environment is osmoregulation, which involves controlling the balance of salt and water within their body. Salmon are able to adapt to both freshwater and saltwater environments, as we've heard, by adjusting the concentration of ions and other solutes in their tissues and by regulating the flow of water across their gills and skin. Another important mechanism used by salmon to regulate their internal environment is thermoregulation, and that involves controlling their body temperature. Salmon are able to maintain a relatively constant body temperature through a combination of metabolic heat production and heat exchange with the surrounding water. And as we've heard from Professor Whelan, in addition to those physiological mechanisms, salmon also exhibit a range of behaviours that help them to regulate their internal environment. So for example, they might seek out areas of the river or ocean with the appropriate temperatures and salinity conditions, or maybe adjust their swimming behaviour to conserve energy and avoid stress. Overall, the ability of wild Atlantic salmon to regulate their internal environment is essential to their survival and success in a range of aquatic environments. We'll further explore the concept of thermal regulation in a future episode of the Higher Biology podcast. So don't forget to press subscribe, and if you've enjoyed the podcast so far, why not give us a rating or follow us on Twitter @BiologyHigher.